Okay, for section 4 here, fourth lecture for chapter 3, we're going to talk internal structures. Now, keep in mind, the majority of bacteria are going to have a singular circular chromosome. Okay? Single circular. Whereas we, eukaryotes, are going to have multiple chromosomes. We humans, multiple straight, linear chromosomes, 23 pairs. Other animals, other plants, other eukaryotes can have more or less than us. Keep in mind, contrary to us eukaryotes, prokaryotes have no nucleus. Okay? Their singular chromosome is just going to be found in a region of the cell known as the nucleoid. The chromosome's just kind of packed up there, out of the way. Our chromosomes, our genome, us eukaryotes, measured in the billions for us mammals, for many plants, measured in billions of nucleotides, B. Bacterial chromosomes measured in millions, M. They also have secondary DNA structures known as plasmids. Plasmids are tiny, small, circular things of DNA that are going to be measured in thousands of nucleotides. Remember a couple of lectures ago, we're talking conjugation, the quote-unquote mating between bacteria that fuse their pilus, the pili. Plasmids are generally what's going to be shared, passed back and forth. Plasmids carry resistance genes, resistance to antibiotics. Got to remember, bacteria and fungi have been fighting it out, making different types of toxins and enzymes, and then coming up with counters or defenses. The toxins, the enzymes that many bacteria have made, many fungi have made, we've co-opted. That's where we get most of our antibiotics. We didn't design them, Mother Nature did. If you're going to have something that's been around for a few million years, there's going to be a defense for it that's also been around for a few million years. Hence, antibiotic resistance. Now, inside the bacterial cell, there's going to be ribosomes, okay? These ribosomes are pretty much ubiquitous. They're relatively the same throughout bacteria. They're quite similar to our eukaryotic ones. Large subunit, small subunit. Prokaryotic ones are going to be smaller. It's not going to have as many components, not as many proofreading abilities, and things like that that our ribosomes have. But all in all, they function relatively the same. Now, with prokaryotic cells, you're not going to have organelles. Okay, no internal membranes and things like that, no Golgi, no ER. But you might have things that are known as inclusion bodies or microcompartments. Inclusion bodies are a storage mechanism. If a bacteria overproduces a certain enzyme, a certain toxin, a certain whatever, it can clump all these together in a certain region of the cell, and it'll form a crystal-like structure. That's an inclusion body. Inclusion bodies can also have metals. Certain bacteria take up metals. It allows them to have a means of recognizing different fields, different magnetic fields that could be around them that would give them an indication of what's going on in their external environment. Microcompartments, relatively new thing we've been talking about in microbiology Here, these microcompartments are going to be little small vesicles, not organelles, just little small membrane bubbles where certain things get stored away. Okay, now we're going to talk about a very important internal structure known as the endospore. Endo means within, spore is a structure. Notice here on the right, this endospore is made within the bacterial cell itself. This is different than spores you're going to see in fungi, in plants. Those are structures that are made outside of a cell, and they're made for spreading the species out into new environments. Whereas the endospore for bacteria, I like to use the analogy of a panic room. A panic room is a special room inside of a house, inside of a building, so that if anything bad's happening, everybody can run to that room, seal it, and then they just wait for whatever bad to go away. Once it goes away, they come out safe and sound. That's what the panic room is. It's a means of protection and potentiating the species, not spreading it like fungal spores. It's protecting the species so that whatever hostile environment goes away, or the hostile environment could be too much radiation, too acidic, no nutrients, water has gone away. Well, once the water comes back, once the nutrients come back, once the acidity goes back to a somewhat quote-unquote normal range, endospore opens up, out comes a cell. When you look at it, as you see here in this figure, a bacterial, they call it vegetative cell. A vegetative cell is metabolically active. It's going through, taking up nutrients, making stuff, breaking stuff down. A signal from outside, one of those signals, water going away, nutrients going away, blah, 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 will trigger the chromosome to duplicate and proteins to start pulling in the cytoplasmic membrane. Proteins will separate the two copies of the chromosome, and then the plasma membrane comes together, and you now have literally two cells. This newer cell, that's the forespore, has ribosomes in it, has polymerases in it, has some amino acids, some stores of things. But most of all that still here in the vegetative cell. This cell here, the main cell, the original cell, still metabolically active. We see that as the endospore begins to form, layers upon layers of different proteins, different polysaccharides, different crystalline structures are laid down in layer upon layer upon layer. Do we know what the different layers are for each of the bacteria? Yes. Do you need to know? No. Just know that multiple layers of different things are there as protective coatings. One coating is good against heat, another coating is good against radiation, another one's good against chemical attack, and so on. It will get to the point that once the endospore is formed, the vegetative cell dies. Chromosome breaks down, the cell wall breaks down, and all you're left with is a spore. That spore is metabolically dormant. Nothing is going on in it. No making, no breaking, no metabolic activity whatsoever. Well, the stressor goes away. Out comes the new cell. Now, these endospores are important medically because we don't consider things to be sterile until all the vegetative, remember these are anything that's metabolically active, it's considered a vegetative cell. All vegetative cells are killed, all endospores are irreversibly damaged so that they can't germinate. So they can't open up and a new cell comes out. Now, when you think medical significance, so what? What are those spores? So no big deal. Well, anthrax, Bacillus anthracis. You don't actually inhale Bacillus anthracis vegetative cells. You inhale dust. You inhale dust particles that have the anthrax endospore on it. The endospore hits your lungs, gets down, bronchi, bronchioles, into the alveoli. Well, that's the perfect environment. It's wet, there's lots of nutrients. That spore opens up, germinates, out comes a new Bacillus anthracis cell. 1 becomes 2, 4, 8. A couple of days later, you've got hundreds, thousands, millions. You have anthrax because you breathed in spores. Also, Clostridium botulinum, or Clostridium genus, and the different species. Tetanus, which is a spastic paralysis caused by Clostridium tetani. You ingest the spores. This was a big problem early on with home canning. Home canners weren't able to get the temperature up high enough pressure to not only kill the vegetative cells but destroy the Clostridium tetani spores. People would go and eat it. They would eat the they would eat the spores. They would or the spores would germinate in the can. And they would eat the tetanus toxin, or they would eat the tetanus spores and it would germinate in their stomachs, and away they go. Turns out this is why they don't want you, pediatricians don't want you feeding newborn babies honey. The honey gets Clostridium tetani spores in the honey in the beehive from the dust. Baby goes and eats it. Its stomach isn't acidic enough to kill the spores. It germinates. Next thing you know, baby has tetanus. Clostridium botulinum, again, home canners. Here, though, this one, it's not you eating the spores, it's you eating the botulinum toxin. Because the spores, once the can's settled down and cooled, the spores germinate, out comes the new cell. It starts making the botulinum toxin and just filling up the can. You go, you eat. Next thing you know, you can't move your arms, your legs. Botulinum toxin is it's paralysis. It's a limp paralysis. C. difficile. Clostridium difficile causes what's known as C. diff. People eat some foods that weren't sterile, weren't properly prepared, has Clostridium difficile endospores on it. It goes, makes its way through the stomach, germinates in the small and large intestine. Next thing you know, they're having paralysis problems, no peristalsis in the gastrointestinal tract. And lastly, Clostridium perfringens. This one causes gas gangrene. This one's not an ingestion. You're not eating this one. You get poked with something. I read years ago a clinical report. Guys riding a horse through the woods gets jabbed by a branch. Didn't go very deep, very little blood, didn't think anything of it. Just put a Band-Aid on it and off you went. Well, it turns out there was dust, there was dirt on the end of that branch that jabbed into him, and on the dust, the dirt, were Clostridium perfringens spores. They were introduced below the epidermis, into the dermal layers, into the fat layers. Germinated, produced toxins. Toxins kill cells. Next thing you know, guy's leg and his foot, bottom half of his leg and his foot, is turning black. Gangrene. Gotta love it.